Hello, everybody. I'm grateful for the opportunity to present my group's work here, and I will use slightly different terminology in my talk. Instead of multi-layer, I'll call them dynamic networks, and I'll explain the difference in terminology throughout the talk. And definitely, I want to thank out of my lab members, Khalik and Jake, who did all of the work that I'll be presenting, and Jake especially for creating the slides on top of which I just embedded some of the customizable changes. So he saved me lots of time there. So this talk is on the task of protein structural classification, which is also called protein fold recognition. So we're dealing with the problem of protein folding, but I promise you this is a network science talk, so I hope that you will benefit from the approaches discussed. And I'm going to start by introducing the problem statement and some of the related work. So the key idea here is, given a protein, or I should say given a set of proteins, extract some features about each protein, be it structural sequence features or 3D structural features, and then train a classification model to evaluate whether we can correctly predict structural classes. Typically, this means cath and scop labels of those proteins. And if you can do that correctly, then if you have a protein whose structural class you do not necessarily know, you can apply your classifier, your trained model to do so. And if you are not familiar with protein folding, what cath and scop databases contain, and I will come to that once I go into methodology, they're basically trying to describe a protein as alpha mostly, beta mostly, alpha, beta, mix, and such. So the goal here is, if you are given a protein, can you recognize correctly what kind of fold it has? Okay? And this is important for numerous purposes in the protein folding community, but also in the protein functional prediction community, because we know that 3D structure correlates with function. So if you can correctly predict structural labels, then you should be able to correctly predict functions of proteins. In this talk, we are not going to go into the function part. We're going to be stopping with the structural classification part. And I already mentioned traditional protein structural classification approaches extract features from sequence, amino acid sequences of proteins. Obviously, proteins fold in 3D space, so you can extract features from 3D structures. But what my group has been doing for close to a decade now, along with other groups, is first model the structure, 3D structure of protein as a protein structure network, in which nodes are amino acid residues of a given protein, and you link two amino acids by an edge if they are close enough within the 3D fold. And we've done a series of studies, I'm going to comment on them shortly, showing that if you do this and then use features extracted from the protein structure networks to classify proteins into cath and scop classes, structural classes that outperforms traditional 3D structural and sequence approaches. So humor me for a second and say, okay, protein structure networks are the way to analyze 3D structures of proteins. And let me tell you what kind of PSN, protein structural network, I'll just be using acronym from now on, PSN, what kind of PSN approaches exist? Or I should say PSN types. So if this is my toy protein with 4 amino acids, and that's the network, PSNs can be ordered or unordered. Unordered simply means we ignore any order of amino acids in the sequence, meaning nodes have absolutely no order information. But if we account for a relative order of amino acids in the sequence, in this case we have 4 amino acids, so there will be just order of 1, 2, 3, 4, then our PSNs would be ordered. They can be unweighted or weighted if we don't account for any spatial basically, traditional approach is if 2 amino acids are within a given cutoff, typically 4 to 6 angstroms, these are tiny 3D distances, then you put an edge between 2 amino acids and that's it. Every edge, every pair of proteins that's below that cutoff gets an edge, and you don't assign any weights to those edges. But you could assign weights, and traditionally the approach, popular approach is amino acids that are far apart in the sequence, but close in space, would have high weight, would be important, because that indicates that they're folding close by, not because they're close in sequence, but because of some folding properties. So if you do something along the lines, you would end up with weighted PSNs. And depending on different combinations of these parameters, Original PSNs have been unordered, unweighted, and that's our paper from 2017, and that's how people have traditionally modeled PSNs. Ordered PSNs are ordered, although unweighted. Weighted are unordered and weighted, and there is no currently ordered weighted because it's just exponentially many possible things you have to account for. So that does not exist yet. And now, if you're still not convinced that we need to model 3D structures as networks first, here's why. In a series of these studies, all of which are in the task of protein structural classification, we have shown that our approaches, starting with original, then we proposed ordered, then we proposed weighted PSNs, along with approaches for analyzing them, that they outperform traditional sequence and 3D structural approaches. Representative ones are listed there. Moreover, different groups have shown in other tasks, including protein function prediction, that if you first model 3D structure as a network and then use deep learning, namely graph convolutional networks, that outperforms traditional protein function prediction approaches, including those from Kappa Challenge. And moreover, they've shown in the last year's ISMB talk, the same group whose paper I listed there, that if you use graphlets, don't worry yet what those are, I'll mention them in a moment, to analyze, to do protein function prediction, that's even better than using graph convolutional networks. So that's why in my talk, I'll focus on graphlet-based approaches for analyzing these networks, but I'll come to that in just a moment. The key point of this talk is, okay, all of these networks that currently exist, these PSN types that currently exist are static, but proteins fold dynamically. Protein folding is a dynamic process. The generation of 3D structure is a dynamic process. So can we do something about this? Noting that we don't actually have these protein folding intermediates available, obtaining that experimentally is virtually impossible. Maybe you can do it for 1 to 2 to 3 proteins, but not for hundreds and thousands of proteins. But still, even if we have the final 3D structure available, can we somehow model it dynamically? And that's what we are doing as our next step that I'll be talking about today. So basically, rather than taking the final 3D structure and model it as a static PSN, can we somehow model it as a dynamic PSN? And I want to emphasize again that the two static versus dynamic PSNs are the different models of the same data. They're using the exact same information, okay? So just keep that in mind. And just to explain you in hopefully very vague terms so I don't confuse you, what we do is given a sequence of a protein that folds in 3D space as illustrated here for a 15 amino acid toy sequence, we take k amino acids at a time and build in the sequence and build our network. So say that k is 5, I would take first 5 amino acids and build the first snapshot of my network, my dynamic network, the next 5 amino acids. So now that would be amino acids 1 to 10 total. The second snapshot, so the snapshot is cumulatively growing. And finally, I would have amino acids 1 to 15 as my third snapshot. 3 is just an illustration. We have typically a lot more snapshots in our networks. We've tested several ways to construct dynamic networks, and this simple approach worked quite well. And 5 is the actual number we use in our paper. It just kind of corresponds to alpha helices and beta sheets in terms of the folds. So there's biochemistry reasons why 5 seems to work, but it's a parameter and definitely can be played with. So we call the sequence of these, the set of these snapshots a dynamic PSN, but then the reviewers who were possibly coming from protein folding community said you are not actually capturing the dynamics of the folding process because you're still dealing with the final PSN, final 3D structure of protein. So that might confuse, using the word dynamic might confuse the reader. So we did agree in the paper to change it to multi-layer versus single layer PSNs, but just for the purpose of the talk, I'll call them dynamic PSNs because we are dealing with dynamic networks, just not process dynamics of the folding process. So now we introduce another PSN type called dynamic networks, and we just, compared to original PSNs, we still treat them as ordered and unweighted, just introduce dynamics. If we can show, so let me do one more click and then I'll explain this. So what I've highlighted in each row, original, ordered, weighted, is in green is where a given PSN type matches our dynamic PSNs, and in red is where a given PSN type mismatches. So dynamic PSNs differ from original ones in just the static versus dynamic component. And all that's needed to show that dynamic PSNs are valuable is to show that they outperform original. That would be sufficient. If we can also show that they outperform ordered and weighted, which use more information than dynamic PSNs, they use information of order of nodes and they use information about weights, then that would motivate the need to develop ordered or weighted dynamic PSNs in the future. This is just the first step. And very briefly on the methodology, the hypothesis is adding dynamic information into the PSN should maybe, hopefully, yield a higher protein structural classification accuracy. And we use data from protein data bank to extract information about protein domains. So our objects are not entire proteins, but something called protein domains, where protein domains are those labeled by Cath and SCOP labels. And these are hierarchies that contain increasingly detailed classes. And I'll try to illustrate this using dummy class names to help explain things. The actual class names are different. So at the primary level of each of Cath and SCOP, there are 3+ classes that say, okay, this protein domain is mostly alpha helix based, mostly beta sheet based, or a mix of the two. And each of those, for example, alpha has alpha 1, alpha 2, alpha 3 at level 2, so does beta. And each of those has alpha. For example, alpha 1 has alpha 1, 1, alpha 1, 2, alpha 1, 3 at level 3, and so forth. So each of these green boxes we have highlighted, we call it a PSN set. Each protein domain that has level 1 label has level 4 label as well. It's just what level of info do we use. So in a given PSN set, for example, if you're studying this primary PSN set, for all proteins under consideration, we're trying to distinguish between those that are labeled as alpha versus beta versus alpha beta. At this alpha 1, 1 bottom left green box, which we call PSN set, we are only focusing on protein domains labeled as alpha 1, 1, and we're trying to distinguish between those that are alpha 1, 1, 1 versus alpha 1, 1, 2, and so forth. And in total, we are studying 72 such PSN sets spanning 44,000 Cath and SCOP protein domains. And we need features and we need a classifier. So the features that we use are graphlets, because graphlets have been super popular in studying various types of networks, including protein-protein interaction networks, and also protein structure networks. And also remember the paper I pointed out that showed that, well, a talk from last year's ISMB where they've shown that graphlets can outperform even deep learning on graphs. So we believe they're very useful, and more importantly, we have original graphlets, which we can use to study original PSNs. They're illustrated on the left. So given a toy network, as shown, for each of the graphlet types, illustrations are listed here for 3 and 4 node graphlets, which count how many times the given graphlet appears in your network, and that's the feature vector for a given network. And original graphlets are again dealing with unweighted, unordered networks, and then you have ordered graphlets, weighted graphlets, and there are even dynamic graphlets. All papers are co-authored by me or coming from my lab. And then given features of the protein structure networks that we have, we do five-fold cross-validation, and using simple logistic regression classifier for this purpose. We tried SVM, didn't do anything other than add extra time. Deep learning is definitely something to explore potentially in the future, but as I will demonstrate with dynamic graphlets, even with simple logistic regression, we can reach for majority of 72 datasets close to perfect performance. So features is what matters technically. And we measure performance in terms of misclassification rate. So for each of the 72 PSN sets, we count a number of PSNs classified incorrectly over total number of PSNs. So the lower score is better performance. And finally, results. What you see on x-axis are the 72 PSN sets. They're ordered in a certain way. Let's not go into those details. Lines technically don't mean anything other than to give us an idea which PSN type outperforms which other if the signal is clear. And again, y-axis is misclassification rate, so lower score is better. In red are dynamic PSNs, and we need to show that they're better than blue. That has to happen to justify the use of dynamic PSNs. And we see that to be the case, but they also outperform very often weighted and ordered graphlets, and I'm going to summarize this numerically. Dynamic graphlets outperform, dynamic PSNs outperform original ones in 64% of the cases, are tied in 4 cases, and are inferior in 4 out of 72 cases. And dynamic PSNs are statistically significantly better than original PSNs after adjustment for multiple hypothesis correction. And similar statistics for the other 3 PSN types hold as well. So we can see that not only are dynamic PSNs better than original ones, which we expected, but we had to verify it, but they also outperform weighted and ordered ones, which means each piece of information adds something. Ordered and weighted help compared to original, but dynamic helps further. And if the more information we can include, it seems to help. So there definitely seems to be promise of dynamic analysis of protein structures. And the goal is to get those actual protein folding intermediates, because once first 100 amino acids of a protein fold, once the others come out of the ribosome, that could change the folding of the original 100 amino acids, and we currently have no way to capture that information. But when we're going to get there, if ever, that's not up to us, that's up to experimentalists, so we could be a year or 10 years or who knows how long away from that. This is the best we can do to try to approach anything related to protein dynamics. And just advertisement by my group, if you're interested to moving from protein structural classification to protein function prediction. In our recent paper, we have basically modeled, kind of touches on this, but extends also on the data that we've used. Here, what I'm showing is on this top level of the multi-scale data is a protein-protein interaction network where nodes are proteins, and you can use the protein-protein interaction network to predict functions of proteins, but each protein, as I just illustrated, is a structure that can be modeled as a network. So now we have a network whose nodes are networks themselves. So this we propose this idea of a network of networks, and we actually use it to predict functions of proteins in humans. So if you're interested in that idea, feel free to check out the paper. And I want to acknowledge for sure Halik and Jake again, who did all of this work, as well as collaborators on the study. And the paper reference is listed here. I don't know if we have any time for questions, and it's totally okay if you want to go and grab some food. I'll be here, so please come ask me any questions if you have any. Thank you so much.
Thank you, Tijana, for this great talk. We have time for questions, so please come to the microphone if you have any. Okay.
Hi, that was a fantastic talk. So the performance was fantastic on the last couple of slides. That was really great. There was just a few examples there where the dynamic wasn't as good as the other ones. Have you looked at the examples where it fails? And is it just, you know, those proteins are just a little crazy, or just, I'm just curious about looking in depth in some of these, and can you learn from
we
those?
looked, are they among alpha proteins? Are they among beta proteins? Or are they at the given level where the data, where one method fails? We couldn't find any sort of signal that's like,
Okay.
We don't understand why it doesn't seem to be. Sometimes it's for alpha at level 3, sometimes for beta at level 1.
Okay.
There's no pattern that we could detect, but we tried.
Okay, all right,
Yeah.
great, thanks.
Hi, wonderful talk. I was wondering, have you looked into any of the proteins that have more dynamic folds based on things like partner molecules or solvents, using PSNs for, I guess, stuff like intrinsically disordered regions? How well do PSNs work?
We are not dealing with anything disordered. We are dealing with data that's in PDB, which is strictly known to have structure. With biochemistry collaborator Patricia Clark, we are interested in many of those questions, but short answer, no, we have not yet.
Okay.
Yeah, thank you.
Cool. Thank you.
Hi. Really interesting talk. Thank you. So have you had a chance to look at trying to make your subgraphs from the other side as opposed to the...
Yes, and believe it or not, it didn't seem to produce any difference.
Great.
It was still
that
superior.
Makes sense.
So that was our, like, if we can show that it works one way but not the other, it would mean something different. Than, yeah, it works both ways just fine. So it's the information that's contained in these snapshots, no matter which way you go, that can provide. Yeah, so it's just more detailed model compared to static PSN.
Okay, because
but it's not the dynamics of the folding process. For that reason, we cannot claim that.
It's not capturing that. Okay, got it.
Thanks.
Hi, thanks for the talk. I was wondering, these ordered PSN models, shouldn't they already include some information about the dynamicity of the protein synthesis? Because if you know the order of the amino acids, you know in which order they are added to the nascent protein.
Yeah, they do. I would argue just a slightly different view there. The dynamic networks all include some order rather than ordered including dynamics. But I think both ways it holds. You're right, absolutely. But still, it's the case that they are not identical, which is fine. It's a different methodology. But yeah, you're right. That's a great. I was planning to mention that, but it would take me 5 minutes to explain it. So yeah,
Okay,
great observation.
thanks.
Hi, great talk. I like how you're using amino acid sequences to construct the network. Have you tried to use the amino acid affinities or the binding affinities to add weights to the edges to
What exactly do you mean by affinities?
like how the amino acids kind of, even if they're far away, they can be near together in
Yeah, so
3D structure.
we are basically using the weights as a combination inverse of two things: how close things are in sequence and how close things are in structure. And things that are far apart in sequence but close in structure are weighted the most as most important. And then those that are close in sequence, which guarantees they'll be close in structure, are the least important in our. It's our heuristic for what we are interested in. Depending on the question, you might weigh things differently, but we are. That's exactly what we're doing.
Okay, thanks.
Yep. Thanks.
Hi, I really enjoyed your talk as well. Thanks. I'm just wondering the embedding you used for the networks. Are you aware of any use of that type of embedding to predict PPIs?
We're not using embedding. We're just modeling the data trivially. So we're not dealing with embedding as defined in machine learning world. But people have used structural folds to predict structures, but I'm not yet aware of using PSNs to predict PPI, to predict interactions, but I'm not yet aware of using PSNs to predict interactions, but meaning PPI, protein-protein interactions. Have we thought about this? Yes. So it's like how lower level Lego-like patterns make higher level. Yeah, we thought about it, but I'm not aware of a paper that exists out there that has done it. So that's awesome idea. Yep.
Okay, thanks.
Thanks.